The first speaker is Matthew Merritt. He's a faculty member in the University of Texas Southwestern Advanced Imaging Research Center, and his title is Hyperpolarized MR Probes.
I'd like to say thanks to the organizing committee for inviting me to speak. It's a pleasure. No conflicts of interest. I'm going to start out and just give you a very quick summary of the physics of DMP, and then I'm going to go into the way I've tried to frame this talk is actually talking about pathways and disease processes, and how you can use hyperpolarized MR probes to get at certain specific questions. And it's a little bit different way to organize things, but I think it's informative. This is the slide that is the root cause of everybody making fun of magnetic resonance. And it tells you why our sensitivity is so bad. In a standard spin-a-half system, the energy that you have here between the transition that you're trying to measure is exceedingly small. And if you look at the equation here, so this is the magnetic energy over the thermal energy, the problem is that, say you've got an Avogadro's number of spins, just from the top, you take a 10 to the 6 haircut for the number of spins that you can actually observe at anything approaching a regular magnetic field strength. And therefore, with the temperatures that you have at 37C, every population difference except for an extremely high field for protons is on the order of parts per million. And that can be a big challenge for sensitivity. The only reason that proton MRI works is because you're 110 molar in protons in the water. Now, one thing that I want to mention is this T right here. Colder implies that you've got a larger population difference, and that's not really particularly useful unless you can do the type of experiment, dynamic nuclear polarization, that I'll talk about right here, and then show the seminal paper on the next slide. If you get down to something that's actually reasonably achievable, about 1 Kelvin, which you can do with liquid helium in a powerful vacuum pump, if you take an electron that has a magnetic moment that's about 660 times that of proton and a reasonable field strength, you're polarizing the electrons at something close to unity, just from the advantage that you get with the temperature. Unfortunately, to do something similar to that for protons and carbons, you have to get into a millikelvin range, which is only accessible through a helium-3 dilution pump, and that's really not practical for any noticeable subset of the population of medical researchers. The key realization that's actually been around since the 1950s was that if your nuclear and electronic spins are dipolar coupled, then a radiation at the electronic frequency causes the nuclear spins to take on the population characteristics of the electron bath. Therefore, you go from parts per million up to, actually, you can get 40 or 50% polarization, which means you've got an enhancement of 10 to 50,000 times. It's pretty amazing experiment. The key realization was that there is a way that you can use this in medical imaging. This phenomena had been used in particle physics for a long time, but in 2003, Jan Henrik and Klaus Goldman came up with this idea that you could polarize it just like a standard experiment, but then you could rapidly dissolve the experiment by putting in boiling H2O and make a hyperpolarized imaging solution that you could inject. Just want to say, for people that are in here that are maybe trying to learn some things that are using PET or some other imaging modality, we are T1 limited, which means that there's always this restoring force that's trying to take our polarization back to the Boltzmann equilibrium, but that is not necessarily a showstopper because, for example, pyruvate, which is what I'm going to talk about mainly today, you've got about a 2-minute window for study. The thing about MRI imaging is that from the very beginning, people have been working on accelerating the experiment, and you can actually do very fast imaging and still take advantage of the increased polarization. If you just want to dissolve the sample and inject it straight into a research animal, you can start that 8 to 10 seconds after the dissolution. The point that I really want to make is that no bonds are broken or formed. If you want to use this in humans, you've got to clean the sample up, but you don't have to make a new sample. You're going to have to get the radical out of it. So you put a tridal free radical into the sample, but it's kind of greasy, and it's relatively easy to actually get out, and I'm going to show some results in humans. Okay, so that's kind of why we do dynamic nuclear polarization. We've got to get our sensitivity up in order to see carbon-13 or another nucleus. Now I want to talk about the disease processes and the pathways that we're interested in, and that we've actually used this technology to look at so far. Now, the Warburg Effect, in the 1920s, it was observed that most cancer cells produce energy by glycolysis and lactic acid fermentation rather than oxidation of pyruvate, and that they can have these dramatically increased rates of glycolysis, and that can be the foundation for a lot of things. F18 PET, you've got a glucose, you've taken off a proton, put on a F18. The fluorination prevents the forward metabolism of the glucose, therefore it builds up in the tissue of interest, and so you're looking at glucose deposition in the tissue. Obviously the sensitivity, even with all the hyperpolarization, far outweighs anything that we can do, but it does not, so to speak, show us downstream metabolism because we're just looking at the radioactivity, not at any of its products. Now, we can do something very similar to that in hyperpolarization. In this case, this is some very nice work done with uniformly deuterated, uniformly C13 labeled glucose, actually in yeast cell culture. In this experiment, the deuteration, the reason we've put these deuterons on here is to try to increase the T1 of the sample. It polarizes well. The yeast cells are actually highly fermentative, and so if you put the glucose in, this is actually still one of my favorite figures that I've ever seen in a paper on hyperpolarization, because you see every glycolytic intermediate. The glucose goes in, you see the dihydroxyacetone phosphate, you see the lactate, you see the bicarbonate that's coming from PDH flux. I mean, it's all right there in real time, and this is just a fabulous result. But it really does take advantage of the really fast turnover of metabolism that happens in yeast cell culture. This is an imaging experiment in a mouse using the same compound. This is just a glucose image as a function of time, 8, 12, and 20 seconds. This is a recent paper, and so this is glucose uptake. This is kind of an analog to FDG PET. They didn't see any downstream intermediates in this paper, and at the ISMRM, Kevin Brindle actually had multiple results showing that you could see lactate after you put the glucose in, which is a really incredible result, but it's not something that's appeared in the literature quite yet. Now I'm going to talk about pyruvate, which is the end product of glycolytic metabolism. I'm going to spend most of my time talking about pyruvate. Usually what I hear is somebody comes up and they're kind of semi-interested in DNP, and they're like, aren't you tired of pyruvate? Isn't that boring? It's not, because there's so many different ways that pyruvate can be metabolized. And here's just kind of a schematic for pyruvate metabolism. Right now, we're just going to look at the red box. You can see that through lactate dehydrogenase or alanine aminotransferase, it's in rapid exchange with alanine and lactate, and this goes right back to the Warburg effect. And this is the first human imaging paper from the UCSF group, and this was a cohort of patients. It was a phase 1 clinical trial, and they got what I thought was really compelling data. Here you see the proton spectroscopy that they're trying to do on the same regions of interest that they're doing the carbon-13 CSI in. You can see that the signal to noise is really pretty close between these two imaging modalities. They can track the lactate exchange with the pyruvate and get a forward flux and rate constant out of that. And the point that I want to make about this slide is this is the worst it's ever going to be. You know, the alpha polarizer that they were forced to use at UCSF, they had to go through, use the pharmacist to have an extensive QC process before they could inject it into the human. And literally, the injection of the pyruvate started almost a minute after it was dissolved. So a lot of polarization was sacrificed before it was even injected into the person. That's only going to get better. And so the sensitivity in general is just going to increase. GE is now marketing a automated QC system that takes care of this and really speeds things up. And so this is the first study, and this will be the worst study. And I think it is still very compelling, very interesting data. Now, pyruvate, aside from the Warburg effect, all kinds of tissues consume pyruvate pretty much as much as they can get their hands on. The heart, for example, uses pyruvate exceedingly fast. The myocardial TCA metabolism is the primary source of ATP in the heart, and it's pretty amazing that the ATP pool turns over about every 10 seconds. So literally, we produce and consume over 10 pounds of ATP every day in our heart alone, which that's why we run at 98 degrees. We're running hot, you know. And so now we can think about not just exchange into alanine and lactate, but once the pyruvate goes into the mitochondria, it can go through PDH. If you're using one labeled pyruvate, then it knocks off a hyperpolarized CO2. That allows you to do another piece of data that I think is some of the most compelling data that's been produced with hyperpolarization. This is actually a study from 2008, and it's a porcine model of myocardial infarction. The top row here is before they occluded the artery. All the metabolites, lactate, alanine, bicarbonate, look pretty much the same. They occluded the artery right here where this blank spot is, so you can see that the alanine and the lactate produce much the same image, but there's this defect in PDH flux right there where the heart attack was. So this tells you that oxidative metabolism is at least stunned in that region of the heart. That's an extremely important insight. Now, this was done with a standard CSI experiment that's not really going to translate into human use. This could very easily translate into human use. This is multi-slice imaging in the pig. Again, here you can see the pyruvate and the bicarbonate image. This has only continued to get better by using, this was just with a straight surface coil. There's already a paper out where using multiple surface coils to increase the homogeneity of these images. But this is a protocol that I think that could easily be used in humans. We're not done with pyruvate yet. It can be oxidized via PDH, but in the liver, pyruvate carboxylase flux to make pyruvate into oxaloacetate is actually much higher than PDH flux. The liver doesn't want to burn pyruvate. It wants to use pyruvate to make glucose. Therefore, when the hyperpolarized C1 position comes in, it can come in through PC, exchange into malate and fumarate or into aspartate. Here you see some experiments we did in the perfused mouse liver. So you can see this asymmetry between malate, C1, and C4. So we're actually looking in real time at the exchange of pyruvate all the way back to fumarate, which is a symmetric molecule, and it's coming back forward with a decreased intensity. So this is a straightforward real-time estimate of how fast oxaloacetate is exchanging with fumarate. This is a similar experiment from the RADA lab in Singapore, where they did exactly the same experiment and see some really nice observations. So pyruvate has multiple metabolic fates and it's really tissue dependent. If it's in cancer, you're going to see a lot of lactate. If it's in the heart, you're going to see a lot of bicarbonate. If it's in the liver, you've got a different set of observations. Another advantage of magnetic resonance is the specificity of because of the chemical shift that's associated with the innate carbon-13 or proton spectrum. Therefore, 1C13 pyruvate is not the same as 2C13 pyruvate. Here you can see an experiment in a perfused rat heart where 2C13 pyruvate was used, which makes 1C13 acetyl-CoA, and therefore you can see glutamate C5, citrate labeling acetylcarnitine. So pyruvate, in addition to its multiple metabolic fates, by a prudent choice of your labeling, you see different pathways as well. There's other probes of oxidative metabolism. I've been talking about carbohydrate metabolism. Here's an experiment with acetate, and this is the group in Lausanne, and this was really a magnetic resonance tour de force, where they're looking at the rat brain, and due to a lack of difference in chemical shift between the acetate and the glutamate, they couldn't really resolve those two peaks to track them separately. They did some fancy polarization transfer and actually identified the production of alpha-ketoglutarate in the brain. I would not have expected that. I would have expected glutamate. The alpha-ketoglutarate pool you would think would be much smaller than the glutamate pool, but their data is pretty convincing, and that's an example of where acetate can be useful in the brain. Now, acetate skips CPT1. It doesn't need to be transported. It's not beta oxidized. It's just transformed into acetyl-CoA. If you use 1C13 butyrate, you still bypass CPT1, but now you get into beta oxidation. This is another paper from the group at Oxford where they used 1C13 butyrate in the perfused rat heart. And you can see glutamate, beta-hydroxybutyrate, citrate, acetoacetate, acetylcarnitine. So here we have these probes not only of carbohydrate metabolism but also fatty acid metabolism. Unfortunately, I'm not quite sure it's going to work for long-chain fatty acids, because when you bind them to albumin, their T1s get really, really short. But nonetheless, I think these could be very effective tracers. pH, everyone's favorite target for imaging. Specifically, I really like the hypothesis about low pH being how cancer is actually metastatic, and another 200 things that I could put down here where pH is important in dysfunction. You can get at this with straight bicarbonate and the Henderson-Hasselbalch equation. There's a lot of advantages to using bicarbonate. It's really the most physiological of any agent that you could imagine. Unfortunately, it's got about a 10-second T1, so if you're going to use it, you got to use it really fast. It does not have the T1 of Pyruvate. This is some work that we did at UT Southwestern, and I just want to make the point that you're not restricted to carbon-13, and two, there's different advantages to different nuclei. Yttrium, for example, has a T1 that is much longer than any carbon-labeled compound that's out there, and therefore you can hyperpolarize this. And here you see the titration curve for DOTP, and so extremely long T1, and therefore there's some advantages that to that, as well as an extremely long T2. And EPI could actually, I think, be quite useful for yttrium because it actually has about a 15-second T2, and you could get a lot of echoes out of that. Now I want to think about diet. This is a really stunning number. 10% of U.S. caloric intake is fructose. And despite what the commercial is about corn syrup and sugar, well, sugar is not sugar. Fructose bypasses the key step of glucose to glucose 6-phosphate and all the regulation that happens right there. It just goes in and floods the glycolytic chain. This is a paper that's actually from 2009 where they put a C13 label right here at this antimerric carbon. There's no proton that's attached, so it's got a really long T1, and they can see this flux through hexokinase. And I think this could be a really, really useful agent if you're just interested in the impact of fructose on diet in the U.S. Another big problem is inflammation. The inflammatory response is implicated in everything: diabetes, non-alcoholic fatty liver disease. And here's an imaging agent, Dehydroascorbate, that from a spectroscopist standpoint is kind of boring. It's either DHA or vitamin C. It's two peaks in the spectrum, but the point is the chemistry that it gets at, because it's looking at NADPH and NADP and glutathione and how the body handles excess reducing equivalents or excess free radicals in the body, and therefore this is a compound that I think has a really bright future just on looking at the relative peak heights of two different compounds. This is a semi-complete number of enzymes and pathways that you can look at. I talk about beta oxidation and glycolysis in all caps terms. This is just some of them. This list is growing constantly, and we really I think have in a very short period gained a lot of insight into a lot of different pathways. Okay, in summary, I think this technique is going to be useful in basic biological sciences, but I also think it's going to be useful for diagnostic imaging. It's really powerful to be able to see this metabolism in real time. This is not a tracer experiment, unfortunately. You use a lot of compound. Even with the hyperpolarization, you still want to target for pyruvate something like 4 or 5 millimolar circulating in the blood. Now, pyruvate is really safe, and that's something that was well tolerated in the study that UCSF just completed. But at the same time, you've got to be aware that the metabolism that you're looking at is really related to a bolus. You're not looking at a steady state metabolism. So that's not a showstopper, but it's definitely something that you need to be thinking about. Finally, I'd like to thank these funding agencies, and thank you for your attention.